Hello and welcome to Mr. D's Science Videos. In this video I'm going to be explaining genetic engineering. So I've got an example here. Here are jellyfish which contain a fluorescence gene which allows them to be fluorescent, which means glow in the dark. And here are some rats that have been genetically engineered to contain the same gene. So what they've done is they've removed the gene from the jellyfish, inserted it into the rats, which gives them the feature. So we're looking at a simple definition of genetic engineering. All it is that we've removed a gene, which is a piece of DNA for a feature from one organism, and inserted it into a different organism. So this means that that new organism would display that feature. For example, fluorescence in rats. So when do we need to do it? We need to do it at a very early stage of development. For example, a fertilised egg cell. This means that when that egg cell divides, every new cell will contain that gene. If we try to engineer an adult, for example me, we just would engineer one of my cells, but all my other cells would still be the same as they were before. So genetic engineering would not work. So every time that egg cell divides, all those features would be there. So it can be used to engineer and produce organisms that are useful for humans. For example, genetically engineering bacteria to produce insulin. So it's a step-by-step process. Step one, we take the human DNA from a human cell, and we have bacteria there. All we need to do is remove the human insulin gene using enzymes. Now that's key, I mentioned enzymes, you need to know that. So remove the human insulin gene using enzymes. You would then cut out a section of the bacteria DNA. So now we have the human insulin gene and the bacteria DNA. And then, again, using enzymes, insert that gene into the bacteria. So now we have a bacteria which contains a human insulin gene. The next step would just be to allow the bacteria to divide and grow. Every time the bacteria divides, they would all contain that human insulin gene. This means that the bacteria will be able to produce insulin, which can be used to treat people with diabetes. And that's what we'd end up with. Lots of insulin, which can be used to treat people with diabetes. So, another thing you need to know about is GM crops. Now, GM crops are just plants that have been genetically modified. And they could be genetically modified to have these features. Resistant to being eaten by insects. So a gene may allow the plant to produce a chemical that stops the insects from eating them. That could be one example. Another would be resistant to herbicides. So when we sprayed our crops with herbicides, it would kill the weeds, but not the crop that we wanted to grow. For example, corn. Negatives. There are a couple of negatives that you need to know about. They may have a negative impact on insect and flower populations, because obviously they may be killing those insects that will eat the crops, and they may have negative effects on human health. Thank you for watching. Please like, subscribe and share. You can also find me on Twitter at Rushcliffe Bio. Thank you very much.